the Dementia Researcher podcast, talking careers, research, conference highlights, and so much more.
Hello and welcome to the Dementia Researcher podcast. This is the first of two sessions coming to you from the International Lewy Body Dementia Conference here in Amsterdam. Hi, I'm Josh Harvey. I'm a postdoctoral research associate at the University of Exeter, and I'm here today joined with three lovely guests sharing you the insights from the Lewy Body Dementia Research Conference. Obviously, long-time listeners to the Dementia Researcher podcast will know that they cover a number of different conferences over the year, but this is the one specifically focused on Lewy body dementia. For those unfamiliar, Lewy bodies, as they're referred to here, are abnormal protein aggregates in the brain formed primarily of alpha-synuclein, and their buildup is characteristic of a number of different clinical syndromes, namely Parkinson's disease, and of interest for us today, Lewy body dementias, which comprise both Parkinson's dementia and dementia with Lewy bodies, although maybe controversially, how distinct these two are is under vigorous debate. Without any further ado, I'll introduce my fantastic three co-hosts and panelists for this session. I'll start left to right. I'll start with you, Athina.
Yeah, so hi, I'm Athina. I'm a first year PhD student at the University of Dundee in Scotland. I work at Dr. Dave's Koss, which is sitting right next to me. Yeah. And my research is on DNA damage in neurodegeneration, specifically in synucleinopathies. So I'm trying to find a relationship between DNA damage and synucleinopathies, and especially dementia with Lewy bodies, PDD. Yeah.
Fantastic. I guess there's been an initial introduction to your group's work, David.
Yeah. So hi, I'm David Koss. I'm a relatively new PI at the University of Dundee, and primarily, as Athina has already said, we're looking at DNA damage, but actually we're trying to look at a slightly wider scope of that and look to see whether DNA damage is a common theme across synucleinopathies, but also pure Alzheimer's disease, and maybe a few other neurodegenerative conditions as well.
Fantastic. And then last but not least, we've got Joe.
Thank you. My name is Joe Kane. I am a geriatric psychiatrist and I work from Queen's University, Belfast. I do research into lots of different types of Lewy body work, but perhaps most relevant to this conference and to today in particular has been clinical trials. So it's great to be here. It's been a great conference.
Fantastic. So I guess we've got the full remit of research here. We've got core fundamental biology all the way through to clinical facing. Joe, in that last session there, you were sharing some work you were doing setting up a domain rating scale for Lewy body dementia. I wonder if you could kind of give us a bit of background on that work.
Sure. Well, we've bemoaned the fact that in DLB research, we've really relied very heavily on scales that most of the listeners will be familiar with. So your cognitive scales, but also your kind of global scales like the CDR. We've relied on these scales that were really designed for other disorders. We have had more than a sneaking suspicion that these scales just don't work in DLB trials, and that we're maybe missing signals somewhere. So over the past couple of years, really since the last conference, we've been running Delphi studies and we've been developing a new rating scale for Lewy body dementia, and that's called the LBD-DRS. And unlike other dementia scales, it has the full range of symptoms that we see in LBD, and it's an attempt to try and get consensus in the community that this is something we should be moving forward with. I was really pleased with the reception that it got, and it's really motivated us to do more work on this and get it out there.
Mm-hmm. Yeah, it was a fantastic talk, and you mentioned a Delphi consensus there and what the kind of main aim. Could you give us some background on what that actually entails in terms of getting an expert kind of decision on that?
Yeah, really glad you asked that, because it's become such a big important part of the field. It's where you really don't have as robust evidence as you would like for treatment, or for any complicated or nuanced issue. You can get consensus from a group of experts. And it's the same process by which we would have developed the diagnostic criteria, for example, for DLB. What you do is you come up with a group of statements, you send them out to the experts, and you ask the experts to what degree they agree with that statement. And if you get lots of disagreement, it's very clear you either need to change your question or you need to kind of get some kind of balance between two or more camps. And you keep doing that, you keep refining your question, you keep refining your statement, you keep refining your source materials until usually around 70% of people agree with what you're saying. So it sounds reasonably easy, but in a really opinionated, well-informed group, it's tricky. But we're really pleased that people have contributed to so much. So what we expect is we're going to have another few rounds of the survey, and eventually we have at least 70% of people that are happy with it.
Really looking forward to it. And I guess talks weren't the only thing. We were sharing posters and other bits of work at this conference. I wonder if you guys want to go into some of the work you were sharing from your own group.
Sure. So we had two posters effectively kind of highlighting our research in DNA damage in dementia with Lewy bodies, primarily utilizing post-mortem human tissue. And effectively, we've found that there seems to be an elevation of DNA damage in the dementia with Lewy bodies, particularly in the neocortex. And then we've mapped that onto pre-symptomatic young age mouse models of the disease and carried on and done some nuclear proteomics work to investigate those pathways that are kind of up or down regulated in response to DNA damage and repair. We're often actually trying to correlate some of our post-mortem pathology results with as was ante-mortem cognitive scores. So actually your work in trying to find a kind of more accurate representation of the disease burden in patients, whilst I think is probably not your driving motivating factor, would actually be something that as scientists we're actually also really interested in, because then it allows us to try and correlate the severity of our pathology into something that's actually meaningful. You know, often we stay away from things like the, I suppose, MMSE or CDR, because actually that doesn't really reflect particularly that well the symptoms that we're seeing in dementia with Lewy bodies.
And I guess it's always so difficult. You mentioned the post-mortem kind of discovery that you're looking to validate in mice. I guess the snapshot you'll sometimes have at post-mortem from sort of what's available through brain banks doesn't always capture everything and the full complex progression of a really complex disease.
Oh, I mean, without a doubt, yeah.
It's so difficult. I completely agree, having better domains. And like you said, kind of translating it through to animal models as well, kind of getting that multifaceted approach.
Sure.
So you can get insight into that early process. No, great. I guess we'll move on and talk about research highlights outside of our own work. So I was interested in kind of how everyone found the conference, what was kind of some core takeaways and what was kind of most exciting stuff you saw on display.
Yeah.
I'll start with you, Athina.
Yeah, sure. Thank you. So as a first year PhD student, it was my first international conference. It was really exciting to see the talks, the research out there, and it was really interesting for me. And even though it was somehow more clinical than the fundamental biologists that we are, I found it really interesting to see how all of these people work together to bring basically a better outcome for the patients and everyone. So there was a talk that was really interesting. I was really excited when I heard it, and also a poster that somehow contradicts but also agrees with the results. So it was the talk from Shady Rael. Forgive me if I misspelled that. And it was basically the phenotypic version of the isolated Armin sleep behavior disorder using free water imaging. So some of the key takeaways from that, it was how basically free water was increased in the basal forebrain, that indicated basically a higher risk in developing dementia with Lewy bodies. And that was in a widespread, let's say, in the whole brain. So it was widespread in the whole brain. And I also talked with Shannon Chu, who's also working with JD, and they found that, the free water is widespread, whereas, for example, in Parkinson's disease, more located in the substantia nigra. However, when I looked at a poster, that was pretty interesting, and it was from Dunya, she's the last year PhD student at the University of Sheffield. She saw using MRI again, but she thought that she was looking at atrophy though, and she thought that the atrophy was spread out in the Alzheimer's disease, but it was more specific in dementia with Lewy bodies. It was quite the opposite to what they saw with
conflicting
it was
findings,
conflicting. Whereas in the cortical thinning, it was the opposite. So basically, so in dementia with Lewy body, that was more spread, whereas in Alzheimer's disease, it was more located. I can't really remember the specific location, but yeah. So those were the two most exciting things. It was like really new for me as well, since I wasn't working with imaging or. Yeah,
definitely. A lot of really cool stuff in the imaging field. And you mentioned that first study there was around conversion from REM sleep behavior disorder. Was that correct?
Yeah. That's they mentioned DLB and PD. So it was really interesting.
It's been a very hot topic, I think, at this particular one about it as a prodromal symptom to both PD and DLB. Do you think maybe some of those differences between what they report in their free water imaging and maybe some of the other imaging-based studies might be down to sort of when you look in the timeline of pathology, or is it kind of?
So I asked the same question to Shannon. I was talking to her in one of the breaks, and she said that they still haven't looked at the PDD cases because of the really vast and like broad spectrum of these diseases. But she said that they just started looking at these cases, but she sees like the same partner DLB, so they still can't identify what it is. But yeah, I can't really. Yeah, I was.
I guess that's the question everyone's trying to answer.
I can't really say because
How about you, David? Any highlights that come to mind?
Yeah, sure. I mean, just to stay on that imaging thing, I think what was really interesting was we saw a few presentations back to back looking at both in terms of cortical atrophy, but also in terms of changes in FDG, right? So actually brain metabolism. And to me, it seemed clear that there was this massive mismatch between actually where the cells are dying and where there is this hypometabolism that's occurring. So this is an interesting thing because often when we're talking about dementia with Lewy bodies, we say, of course, it's a neurodegenerative disease, but it's not characterized by a huge loss in brain weight at end stage. So it's clearly that it's a very subtle thing that's happening here, and actually the changes in FDG are probably suggesting some form of kind of cellular dysfunction rather than a cellular loss, and that might be, it would be something that'd be really interested to kind of speak to some neuroimaging people to kind of clarify exactly how they explain the mismatch. Yeah,
that's interesting. Yeah, I guess it might speak to kind of different selective vulnerabilities as well, right? If some cells can withstand higher metabolic burden, maybe they survive for longer just because they have that kind of capacity innately.
Yeah, absolutely. But actually more in line with some of the kind of work that I'm directly involved in or interested in. I was really interested in Amanda Woolman's keynote speech. She's from Colorado State. So she was effectively looking at alpha-synuclein as prion strains. She's using both a mouse model as well as a particular cell line. And she was testing really the ability of brain lysates from multiple systems atrophy or dementia with Lewy bodies or Parkinson's disease to effectively induce alpha-synuclein pathology when administered to mouse models. And I think that's something that several groups have been working on. It was interesting in her hands, in her setup, she actually found that it was only really the MSA brain lysates that were able to induce this. And this is already kind of giving rise to that idea that although we're seeing the pathology of alpha-synuclein, the ability of different strains to infect seems to be different between the diseases. But her work was actually far much more nuanced than that, in that she was taking this MSA strain, applying it onto cell lines that were expressing different mutations, and she was able to show that those brain lysates were able to infect a whole host of cell lines, but actually if she then took that sample and put it into a mouse, and then after pathology developed in the mouse, she was then able to take the mouse brain extract and apply it onto the same cells. They'd actually narrowed down their infective profile. So they were only then able to cause seeding and templating in, say, 1 or 2 different cell types. And I think that's really interesting because it was effectively getting to the heart of the fact that the disease itself, either MSA or DLB or Parkinson's disease, probably contains multiple different infective strains, and by putting it into a mouse model, you're effectively amplifying up those that are most aggressive in that system, and they're becoming the dominant species that's present in the brain tissue. And I think that probably holds quite a lot of importance if we're thinking about monoclonal antibody therapies, specifically about trying to target individual diseases or synucleinopathies as a whole.
A kind of one-size-fits-all approach might not be the way to go, I guess, if there's very different and distinct changes that occur with one strain versus another. I also thought that talk was fantastic and thought it was very cool. One bit where a PDD, patient lysate, that she'd found was performing like her multiple system atrophy species. They went back to the pathology and restained, and it turned out there was, if I'm understanding it, glial pathology there, which might explain that different capacity. It's always very cool when there's those kind of back and forth there.
Yeah, I mean, I'm not really sure what the key differences are between her work not seeing amplification with DLB or PD, but for sure, we kind of have this belief that the alpha-synuclein pathology strains in MSA are far much more infectious and behave more in line with prions than say with Parkinson's disease or dementia with Lewy bodies.
I'm really interested to know if that was delivered on the part of the conference organisers. That was the keynote speech on the first day, and it feels like it was almost like a theme throughout the rest of the nuanced discussions throughout was strains.
Yeah.
Now we obviously haven't joined the dots between the kind of mechanistic differences, but it's been very clear to us for a long time that all of our clinical data is really noisy and messy and doesn't seem to add up. And there were more than a few presenters that pointed towards somewhat incongruent results, be it an alpha-synuclein seeding assay result that wasn't expected or didn't fit with our current model of understanding. And the conclusion was, maybe there's something in this strains thing. For me, it was the strains is probably the biggest takeaway from the conference in terms of the conversation that's leapt from the basic science sphere into the translation and the clinical sphere, and we'll be really interested to see how it develops over the next few years.
Yeah.
And actually, sorry, do you want to say something?
No, I just agreed with what you just said with the strains, to be honest.
I mean, what I thought was interesting, specifically when we're talking about that ability to template and misfold in vivo or in a cell system, seems that MSA seems highest. And something that I didn't appreciate is that in the seed amplification assays, it's the reverse. So the MSA cases don't seem to actually seed as much. So, I mean, I know that maybe doesn't necessarily translate to a kind of meaningful thing in the clinic. To us, I think that highlights that definitely there is those individual strains, and it's something that needs to be considered.
Yeah, definitely. And I guess there's been a few talks that kind of look at this from different levels. There's strains, I guess, on the pathological alpha-synuclein side, and there's people who are looking at determining subtypes of disease using a number of different approaches. I wonder in the future, as we begin to kind of align these, whether one subtype might be related to one specific strain and back and forth. It'll be very interesting to see how well these kind of recapitulate each other. Anyway, Joe, what about you? Any specific standouts?
On a similar point to the idea of like different phenotypes and different subtypes, I just found the plenary this morning by Per Borghammer absolutely phenomenal. He was discussing, he's probably more closely related to the Parkinson's world than the DLB, but he was discussing the brain first and the body first hypotheses of PD and LBD, and some of these ideas aren't that new. We've always talked about this idea of body first pathology, seeing pathology in different parts of the peripheral nervous system and migrating up towards the brain, and then that became a bit unfashionable once we realized that not everybody fit into that. And what he did was really set out the evidence so clearly that these are two phenotypes, and he was able to provide not only kind of gross epidemiological figures for, he was saying, 1/3 of people with Parkinson's disease have body first disease, whereas 2/3 of people with Lewy body dementia have body first disease, which is a bit counterintuitive. And then he also put forward this really interesting hypothesis as to why those people who have body first disease seem to do worse. And the big takeaway with respect to cognition and functioning, and the big kind of enduring image for me will be his last slide, in which he put like 4 quadrants up, and one of the dimensions was Alzheimer's pathology, which we're definitely used to discussing on how that influences the expression of Lewy body pathology. But then on another dimension, he had the brain first, body first, and how that might influence 4 entirely different presentations. And that speaks to what we've always seen and what our patients have always told us, is that these are very different experiences and disorders that end up going to very different health systems. But to suggest that they are arising from 2 dimensions in Alzheimer's pathology and body first PD was just fascinating and challenging. And I've never seen so many phones go up and take the grid. And he had previously supported it with lots of data, but he also sent us away with something which was really challenging and made us really think. And actually what was then proposed was that it might end up at six boxes at some stage, but either way it reflects the fact that clinically this is a very heterogeneous group of disorders, and to think that these are all represented by the same pathological process has always been a bit puzzling. Yeah. The other thing that I really liked that he did is he basically put it for his last slide, he did his acknowledgements. His last slide was basically like an infographic. It's like, here's everything I've done in my talk, squeezed in. Yeah. So for all those people that had missed the slide, they got all the slides together in context at the end. So I think I'm going to steal that in the future.
Yeah. I mean, it was a really powerful talk, I think, and it was really, really engaging. I mean, we were both really excited about just the way it was. Just the way it was kind of presented, and I think it's one of these things that I probably need to go away and have a hard think about, really, to really see if I 100% agree with what you're saying, or whether the evidence holds up. But I mean, absolutely brilliant.
It definitely felt like a convincing presentation. Even right at the beginning, where he had those, it was quite a slightly different 2-axis plot, but he had the autonomic symptoms as measured by the invisible heart kind of phenotype. And then the dopaminergic degeneration phenotype along the bottom, and you kind of had those 2 groups already clustering out, even just looking at those 2 dimensions. Yeah. It was very, very exciting.
It's so fascinating as well, because like I did my PhD in that heart imaging, the MIBG, and we were really excited because we thought it was going to be a really good early biomarker. And then we compared it against something like a DAT scan. During my PhD, I compared it just in terms of diagnostic test terms. And the answer was, it's all right, but it's not as good as a DAT scan. So that felt like the end of the conversation. But now we're hearing that this MIBG that we can do, this isn't like RT Quick that is, related to a few isolated labs across the place. We can't do this imaging, and there's a suggestion from his talk that in people with, body first, that 90% of those have an early abnormal MIBG heart scan. And, some of the work from the Newcastle group on prodromal DLB and MIBG and prodromal DLB, it doesn't seem quite just, it was good for ruling in, not great for ruling out. Again, speaks to these are different phenotypes, and for him to crystallize it in the way he did, using the breadth of data, he had AI stuff in there, he had imaging stuff in there, he integrated the genetic markers into that grid. So it was really convincing, persuasive argument.
I was also going to highlight on the data and then all the all the things he used, like you just said, imaging, post-mortem. He used even models like mice models and mice brains. It was really interesting. So like you said, he just had the data there. It's like, look, I know what I'm talking about.
Yeah. Every sort of level backed up.
Yeah, exactly.
It was very, very interesting. Yeah. Back over to you, Athina. Any other highlights from the past few days?
I just want to, well, it's a bit out of the science world, let's say. It was about their carers. So, so there were so many posts, especially today, last day, about the people who were looking after the patients with dementia with Lewy bodies or Parkinson's disease or anything, but especially with dementia with Lewy bodies. And I had a talk with a woman who she actually lost her husband through dementia with Lewy bodies, and it was actually a really, really fast incident. So 2 years after he got diagnosed, he passed away. And it was really interesting to hear how, well, what these people are going through. And it's not just the patient, it's also the carer, because she was saying to me how she was trying to improve the quality of life of her husband. So she was taking him out for a walk, he was trying to play games with him and just to keep him alive, you know. And it was really, really nice to hear, and all the stories and what they do. And also, I think they also conducted a study with carers, and they found out what are we lacking in this sector? So what are these people need? So they found actually that some of the clinicians that are working in this sectors, they've got no clue how to diagnose it. So DLB, they found that there is a lack of support out there. And they found that there is mental instability for the carers as well. It's just not them, yeah. And it was just two old ladies doing this. So that was really, really touching, to be honest, for me. So it was really good to see that it was included in this conference as well. It's just not about the science, but it's also about the people who are taking care of those diagnosed. Yeah.
Joe, I see your...
Yeah, just on that, I don't know if you saw the talk earlier on today from Leah Forsberg in the Mayo Clinic, where a really novel idea of, you know, she built a kind of algorithm that's in a kind of Tinder type way, matched up care partners of people with LBD, to find the right match. And one of our colleagues from Southampton, Jay Ammon, made the really good point that we don't really have good support systems in the UK, at least. No. We kind of give people a little bit of information. We might direct them towards local services, but they're very seldom tuned in to the real challenges of Lewy body dementia. So to have this, which is seemingly working almost autonomously, to link people up with people with whom they can touch base, and it's effectively peer support. And although it does lose a little bit of the human touch in somebody coordinating it, I thought it was just a really inventive way to get people connected. And I strongly suspect that's the type of thing that made a big difference in people's lives.
Yeah, and actually, I mean, if I can just speak a tiny bit about my own personal experience. So I've recently been caring for my mum who's had Alzheimer's disease. And often I've been looking for peer support, but actually being paired up with someone who's at the same stage of my life is actually very difficult, because actually typically it tends to be people that are slightly older, and often it's partners caring for their husband or their wife. And actually it would have been really good for me, I think, if there'd been someone that could have been kind of personalized and selected for that, because certainly going through that process, I definitely was in need of some kind of peer support. Yeah. And so that's why it really struck a chord with me, this idea. And the good thing about the app is it doesn't have to be local. I mean, maybe it would be ideal if it's local, but if there's particular demographics that is slightly rarer to kind of pair up, you can go as far as you want in terms of covering that. So again, it was interesting, and of course they're still looking for participants. But of course you have to be based in the US for this. But
I thought it was really interesting that one of the things that wasn't important in selecting peer support was the distance. You know, and as we've all moved kind of virtually and all more used to communicating via our phones and everything else, you'd like to think it would get even easier for most of us.
Yeah, absolutely. And a really nice, totally different take to give a talk at the conference as well. But I mean, I guess when we were talking about the involvement of carers, and I know there is a Carers Day that's happening tomorrow as well. But you know, I think I think what's been good about the conferences in the past has been that these events not only serve as a hub for us as scientists, but it also serves as a hub and a support structure for those that are undergoing either the disease or caring for someone with these diseases. And I really hope to see that maybe the next time this is happening, that there's a really strong representation for that.
Yeah, definitely. It's essential context, isn't it? It's very easy to feel a million miles away, especially if you're kind of more post-mortem or discovery biology kind of stuff. You can sometimes lack that kind of immediate meaningfulness of what people living with these conditions is like. I thought on the first day there was a really touching moment where someone was presenting around autonomic symptoms and early, I believe it was cardiovascular aspects of the disease, and someone who was a carer came to the mic and spoke to how what they were presenting sort of from a broader kind of research perspective really rang true to their own experience caring for their partner. I thought it was, like you said, it's great to have that kind of mix of researchers and carers in one setting.
And the video they showed as well,
Yes.
the phase wind, I think.
Yeah.
The phase of wind. It was really good. Well, it's what you said about we live in a world that's all digital, so it's really easy to communicate with anyone you want. So it was a group of, it was mostly women who were taking care of their husbands, again, with dementia with Lewy bodies. And there was one of them, she was really disdaining. She was crying and she said, I can't take this anymore. I'm not as strong as you, girls and whatever. But then through Teams, they came in contact, and then they also had a day that was just for them. So they did, so they all come together. They went in a field trip, actually. And then they organized other activities with their husbands. So they all come again together and they started talking. So that was a really fascinating to watch.
Definitely.
So it adds up everything you say as well.
No, I also found that trailer really interesting. I don't know if we can link it in the show notes or anything, but we'd definitely recommend it.
Yeah, that's a good point. I think it's really important my first international Lewy body dementia conference long time ago. But as a first year PhD student at that, it's really easy to feel overwhelmed. There's loads of people here saying, I didn't miss a talk because it touched every area. But as someone new to the field, that can be really, really overwhelming. And I remember what was really helpful and centering and motivating was stepping out of the scientific talks when I got overwhelmed and reminding myself why I was doing it in the first place. So I agree. I think it's great, but I would love them to be parallel. And I know we've done that in previous years, and I would like to see that again in future.
I agree. I think the other thing that we have to talk about is the Rising Star Awards.
The Rising Star.
I can't even remember. So someone I'd never heard of. Congratulations.
Thank you.
Absolutely well deserved. Brilliant.
Really appreciated. And an honor to be included with so many other great researchers. So 3 preclinical, 3 clinical, and a good few of them were sort of presenting and sharing stuff, and it was all really. I was very blessed to be.
I was hearing that you clearly didn't really fancy your chances, because so a friend of mine was kind of looking at the screen and looked over, knowing where you were, and you were still looking at your
I didn't know. I wasn't faking being gracious or anything. I genuinely had no idea it was coming. But very nice surprise, definitely.
Dave, for the podcast, would you say he's a shade of dark red there? Or,
slightly
darker.
But his head has swollen, just a tiny bit.
I appreciate you guys bringing it up because, Clearly,
Yeah.
I'm very comfortable, singing my own praises. Anyway, moving swiftly on. Any other research highlights? Any other core kind of bits that stand out?
So it's not really my specialty, but I was particularly interested in some of the data from GWAS studies. That was, and I believe this is right. I had a problem with a lot of pens at this conference, so my notes weren't actually that brilliant. The free conference pens, that was terrible.
Kept on asking for a pen.
That's
Do you
true.
have a pen?
Wow.
I mean, I could have brought my own. But I believe they conducted it on isolated RBD cases, which again was another common theme of the conference, which is that RBD is almost it's almost a clever way of having a catch-all to start with, because many of them, up to 90%, will go on to develop either Parkinson's disease or dementia with Lewy bodies. So you kind of get in there early and are able to increase your statistical power by the fact that you have this common
Exactly.
goal.
And a real sort of specific signal. I thought that talk was fantastic as well.
Yeah.
This was Emma Somerville, I think.
Yes,
her
yeah,
name is,
that's
from
correct.
Toronto.
From Montreal. Yeah.
Montreal. Right country, wrong place.
Yeah, close enough.
Yeah, I thought it was really cool. Like those RBD, GWAS are really interesting because they flag up a lot of the same genes as DLB and PD, right?
Yeah.
It stand out. GBA, SNCA, always there. But she kind of delved into this new locus, I think it was near this gene RCOR1.
Yes.
And really from like everything she showed from comparing to other GWAS, it seemed to be quite distinct from the signal in PD, DLB, AD. From what I could interpret, it seemed to be kind of a real novel and distinct signature of RBD, which I thought was quite interesting. I guess there's tons of work to be done to kind of figure that out, but really
I mean, it was interesting. I mean, again, we're assuming that it affects this gene, or maybe I am, and so of course, I was just googling what this gene is involved in, and it seems like it's a transcriptional repressor of neuronally expressed genes, which I think is really, really interesting. So it's typically active in non-neuronal cells for this, but I'd be amiss if I said that there wasn't evidence to suggest as a role in DNA damage repair, which I was interested to see.
It all lines up.
And I know that there's perhaps some growing evidence in other genetic studies that are implicating some DNA damage repair pathways or genes as risk factors for Parkinson's under specific environmental settings as well. So it was interesting to see that. And often, actually, having a look at some of the kind of GWAS studies for DLB or for Parkinson's, you often get these genes that are not quite meeting the statistical cutoff point. But I mean, I think at this stage, because we're still so limited in the numbers that we're using for these investigations, there's still a lot of work to look at those slightly near significant hits as well,
Definitely,
right?
Yeah.
So
that was also a core highlight. It was very close to in the previous study, but hadn't quite. And just those extra samples just nudged it over. But agreed.
Absolutely.
Definitely plenty to be delved into. So this is the 8th annual ILDBC, and I think you said this was your first one. I think you were both at the previous one in Newcastle. I'd be interested from you two kind of what's changed in the field since the last one, what kind of stands out as new and maybe not new, and then we can maybe talk about how we think will change in 5 years' time. So first over to perspectives from the last one.
Well, the thing that was really talked about the last time was really cutting edge. Last time it was the first time we were talking in detail about RT-QuIC, about these synuclein seeding assays. And since then, we've learned about data from lots of longitudinal cohorts, from some of the larger consortia. And it's what's quite clear, especially with the fact that it's been kind of commercialized, is when we talk to our American colleagues, they have integrated this tool into much of their routine clinical practice. And they're using it pretty frequently, and they're seeing where it makes sense and where it doesn't make sense. So it's quite nice to see something go from the new kid on the block to see something which is really tested in the field. Now, I would say that there's nice data that's come from it, which, yes, it's sensitive and specific for CSF, but we're surprising lack of specificity, in particular with the skin samples and things like that. So, and you also saw some criticisms of it as well, in that it's a very binary tool. It's a yes or a no, rather than giving you any quantification of the degree of alpha-synuclein pathology. So it's nice just to see those conversations develop. It's nice to see where the zeitgeist has weaknesses, but I don't think it's going anywhere. I do think it's going to become, as we've seen with the staging, the different staging systems that have been proposed. It's probably here to stay for the time being, and it's probably something we're going to have to get used to. So that's the big difference for me, and in a relatively short space of time, that's been a big jump.
Yeah, definitely. It's very present at the moment. It is exciting to see it in clinical practice now. David, any thoughts from previous years?
Well, I'll kind of pick up a little bit from what you were saying about the seed amplification assay, which was that there was also the presentation of a new kind of modified seed amplification amino assay, which was from Omar Alagnaf. And I guess this is a kind of almost has the potential again to be as revolutionary, right? So it has better detection and actually better specificity when it comes to skin samples and various other things. And the critical point of this is that it's actually quantifiable as well. So you can then start seeing, comparing the levels of, I suppose, seeding rather than just this binary yes or no. Yeah. So, I mean, that was one thing that I noticed. I think actually the biggest thing for me was the representation of REM sleep behavior disorder as a kind of isolated phenomenon, and using that as specifically, as you said before, well-defined kind of cohorts to really kind of look at in many different aspects as well. That I don't think that was something that was particularly touched about too much in Newcastle in the last two years ago. Yeah.
I think that's fair. I think previous congresses have really, I was at the congress before that, in which REM sleep behavior disorder was integrated into the diagnostic criteria. It's definitely something that's been out there, but it's the fact they've been able to really, really well characterize those cohorts. Yeah. They've been able to do FDG PETs, they've been able to do MIBGs, they've been able to do the RT quick, the seed amplification assays. And it always feels a bit foreign to me, because it's not something which is well recognized in the UK practice, because it's not really that well developed a specialty. So, sleep medicine isn't there. Yes, I recognize RBD, but it's usually in people who have other DLB symptoms going on as well. Yeah. So, yes, it's really exciting to get exposed to that data, because I personally, the clinic isn't producing any hypotheses around that for me. The data has to.
Absolutely. And I guess the final point, with fresh eyes, I guess, Athina, given this is your first one, where do you kind of think the field's kind of moving forward? 5 years' time, when we meet again for the next, LBDC, what do you think?
Right, so on top of my head, what I can think now is trying to find the difference between PDD, DLB, and trying to, but I think looking from the GWAS studies and everything, I think it's just bringing the different scientists that's out there, for example, fundamental scientists, together with clinicians, whatever, and just trying to find the root cause of these diseases, and also how they spread and where they come from. So, for example, like we said, body first or brain first. So I think that will gain much attention as well. And they're going to keep looking for more, like I said, just the root causes of this spread. I think
definitely. I think integrating it across every single level, and definitely probing into that heterogeneity and kind of.
And especially after the strong data that people showed out there when it came to this, and also because we had the debate. So we had the debate about whether the PDD or the DLB are different or the same, and they should go under the same umbrella, let's say. So I guess that will be the way to go.
Definitely. How about you guys? Future perspectives, final thoughts for where we're going to go in the next 5
Well,
years?
then my experience, so.
I think it's slightly difficult to predict. I see there being more and more emphasis on the fact that for a lot of the cognitive symptoms that we're seeing are perhaps being driven by concomitant Alzheimer's pathology. That seemed to be another kind of common thing. And actually, I think it wasn't necessarily directly proven, but it was just as a kind of thought on it, is that really the presence of amyloid was actually the thing that seemed to dictate whether there was a cognitive impairment or not. And then actually the magnitude of tau pathology seemed to drive that. And I think that's fine in one sense, because I'm quite sure that they're modifying what's underlying this. If it's, synuclein disruption that's potentially causing that, I think that's likely. And then these two things they're kind of adding on top of that to really bring out that cognitive phenotype, if you like. And I guess we're going to see more and more of that. So,
yeah, absolutely.
I'm really excited about. I just think we're working better together. I think from a trials perspective, I think we're better tuned into the conversations about measurement, about characterization of cohorts, and we have better mechanisms of working together and getting in contact with potential participants than we ever were before. And although there was disappointing results from the neflamapimod, the rewind that was presented earlier on today, there was a big recruitment in a short period of time, and it was biomarker enriched cohort. And yes, it used CDR and other skills like that, but I just think we've made strides there in the conversation, and I think we'll continue to make strides over the next few years. Maybe not 2, maybe not 3, but 5 would be really nice to see some better treatments for people with Lewy body dementia.
Yeah, definitely agreed. Lots to be hopeful for.
Mm-hmm.
Well, I'm afraid that's all the time we have time for today, but I'd like to thank our brilliant guests once again, Athina, David, and Joe. If anyone wants to check out talks or look at any of the details of what we discussed, there's a conference website, ILBDC, that you can check out online. I'm sure it'll be in the show notes. And you've been listening to the Dementia Researcher podcast.
The Dementia Researcher podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk.